introduce our next two speakers as well.
Great, thank you, Emily, and thank you to all of the panelists in the previous session. I am really excited to moderate this final session on accelerating innovation through data reuse and sharing. As Emily mentioned, my name is Yvette Seeger, and I'm the Director of Strategic Scientific Program Advancement here at FASEB, so DataWorks is part of that portfolio. And just really excited to have a conversation with our two speakers, Dr. John Chodera and Daniel Harris, to hear a bit more about their projects. So I do not want to take the time away from them, but as with previous panels, they will each give 15-minute presentations, but I will go ahead and introduce our first speaker, Dr. John Chodera, and he will talk about, so he is a member of the Memorial Sloan Kettering Cancer Center's Computational and Systems Biology program, and he'll tell us about ASAP Discovery Consortium, an open pipeline for antiviral drug discovery. And if you had the opportunity to read the information about this Exemplary Achievement Award-winning prize on the HeroX platform, I am looking forward to learning more about this project. So I will turn it over to Dr. Chodera.
Thanks so much for the kind introduction. I'm really excited to talk to you today about data sharing for something where we don't usually talk about data sharing, which is drug discovery. Often it's all locked up in proprietary databases internally to companies, but we have an unusual opportunity to find new ways to share this data to accelerate antiviral discovery globally. So I'll tell you a little bit about that. First, my name is John Chodera. I'm at the Sloan Kettering Institute, the basic science arm of Memorial Sloan Kettering Cancer Center. You can find the slides online here at this URL, and you can find my disclosures as well over there, or go to asapdiscovery.org to find out more about our organization. So what is ASAP? It is the AI-driven structure-enabled antiviral platform, and it is a follow-on to what's called the COVID Moonshot, which was our crazy attempt to make drug discovery open and extremely fast in the face of the emerging and unfolding COVID pandemic back in 2020. So I'll just very briefly highlight what happened there temporally. Everybody found out very early in January, as the cases were unfolding, that it was a new coronavirus that was very similar to SARS-CoV-1. But just a few days later, Shanghai actually crystallized the main viral protease of this SARS-CoV-2 and found it was very similar to SARS-CoV-1, which had been intensively studied many years before. What happened, though, is that they had to shut down very rapidly due to COVID, their own laboratory, and they transmitted the plasmid sequence to Diamond Light Source and Oxford in the United Kingdom, where they were able to express and clone the plasmid and then get crystal structures very early on. And then using the Diamond Light Source and what's called XChem, this amazing high throughput X-ray crystallography facility and fantastic automated beamline, they were able to generate this X-ray fragment screen where a lot of small drug-like fragments fill entirely the binding site and give you a roadmap essentially to developing new therapeutics. We weren't certain anybody would really follow up on this data to go discover a new drug. So we put everything online, shoveled it on through Twitter and web pages. It was pre-printing at this time, and then started this essentially crowdsourced idea of how we merge these fragments into potent initial hit compounds, allowing everybody to submit their own designs. And we quickly were able to make a bunch of molecules, find some really great hits as the starting point for drug discovery, and then using more traditional lead optimization and global scale computing, where we got 1 million people to sign up for this distributed computing platform called Folding at Home. You just run simulations on your computer. It's a screensaver, if you can remember what those are. We were able to rapidly mature these into very potent antivirals for SARS-CoV-2. And within 18 months, we had a new candidate for preclinical development that was funded into an accelerated program by the Wellcome Trust and the World Health Organization's Act A therapeutics pillar. So this is hopefully on its way to the clinic for a patent-free antiviral. And in doing so, we published what we learned, but of course, we disclosed all of this data during this to try to help the world research community as rapidly as possible, and learned a lot about how to share data for drug discovery that then serves us very well in this next generation program that I'll tell you about. But two of the main lessons that we learned, which I thought were really interesting, is that we were able to get an X-ray structure of basically every compound we synthesized, and we made several thousand of these compounds, assayed them, generated all of this data that you might be interested in drug discovery. I'll tell you what that data is like. And we found that researchers were very hungry for using this data and exploiting it for either studying studying the virology of this virus, or studying the enzymes, or actually doing their own kind of drug discovery. So we saw X-ray structures being downloaded in real time way before they hit the protein data bank, which is sort of the fair repository for the community there. We saw preprints that we kept updating over time just take off in terms of number of downloads and accesses. And we saw what was, I think, really satisfying is that even commercial drug discovery entities such as Shionogi cite the structures that we released into the public domain as the inspiration for finding compounds in their late-stage pain program for ensitrelvir, which is now approved in Japan and hopefully fast-tracked for US approval for a SARS-CoV-2 antiviral. So it was able to accelerate discovery worldwide in addition to contributing to our own discovery programs. So when the NIH funded us as part of this antiviral program to run the chemistry for more drug discovery programs for coronaviruses, flaviviruses, and coronaviruses to prevent pandemics as part of this initially 5-year, but now truncated to 3-year antiviral drug discovery AViD Center program, one of the pillars of the antiviral program for pandemics, we thought long and hard about how we can make use of open science to really do as much as possible. And this is basically $68 million entirely spent on chemistry and assays. So thinking about how we can capture the value that the NIH puts into this and make sure that researchers around the world can maximize the utility of this data for future drug discovery when we need it, which might be 10 or 15 years from now, which is sort of the interval between the SARS-1 and SARS-2 pandemics. We thought our first priority is to ensure antivirals are developed in a manner that enables global, affordable, and equitable access to overcome some of the issues we saw with the COVID pandemic. We also want to make sure that we can rapidly accelerate development in emergent pandemics, but as much as we can, we'd like to get all of the data out there as quickly as possible, as long as it doesn't compromise those priorities. So what we built was essentially a distributed biotech. We have everything from the target identification, where you figure out what target protein and am I going to try to drug. Then you try to get structures of this with a variety of different small compounds engaged that might be starting points. These are these X-ray fragment screens. Then you rapidly progress these to initial lead compounds that are potent enough that you might start getting some antiviral effects in cells. And then finally, you do what's traditional lead optimization. I'll tell you a bit more about the multiple objectives you're trying to satisfy here, going through design, make, test, analyze cycles until finally you do a preclinical development program to persuade regulators like the FDA that it's safe to put into humans for the first time. We have a huge number of partners that are involved in this. I can't possibly name them all. I'll show a couple of slides at the end, but essentially there's 150 people contributing to this organization in some way or another that are all essential parts of what we're doing here. So we thought about, how can we use the spare time that we had to actually try to get all of this data online, the data that we're generating. We have to take a pragmatic approach here for sharing data for drug discovery, is also learning from what we learned from the COVID Moonshot. First, every output should be a first-class deliverable rather than an afterthought. We should really try to think about all of the other things that we're making that might have value to shave years off of somebody else's discovery efforts. We also want to prioritize the most valuable stuff first, because we have very limited spare time in order to do this. We ultimately want to ensure that all data ends up in a durable repository that conforms to FAIR principles, because you might not need this data until SARS-CoV-3 or MERS-CoV-2 appears maybe in 10 years or something like that. So you want to be able to find it then. So we've identified repositories that will be around for the long term. And we also want to do as much as we can to remove barriers and minimize the friction to using all our data, as well as being very explicit. We already heard a lot about licensing, but being very explicit about licensing to make sure that we remove all of that friction if possible. So you might have seen this kind of pipeline diagram for a drug discovery company. You go to Pfizer or Amgen, for example, and you'll see all of the programs that they're working on. But in our case, the difference is that you can click on every aspect of this pipeline to see what is all the data associated with each of these stages of drug discovery. And I'm just going to take you on a very quick tour for those of you who might not have seen what this looks like before, about what that kind of data is and what it looks like to share this data, at least in our initial blueprint of how we think we can do this. So we have all of the different classes of outputs, which are represented by all these little icons at the top that you can hover over. And this is live. You can just go to the website and play with the data at first you want to figure out, why am I going to try to target this particular protein? So we've done all of the history and reading for you to synthesize essentially a review about why this would be a good target to pursue, what assays are known already, what chemical matter is already known that you might use as positive or negative controls for this, and really any advice that we can have in someone who's starting out to target these as well. The next stage is somebody might just want all of the molecule data that we have available for this. So you can just easily grab that and download all of the assay data in a CSV file immediately after that's always up to date. But in the longer term, we're trying this, trying to get it into a fair repository. In this case, Chembl is one of those repositories where everybody looks to pool data. It just takes a lot of effort to get into these databases as well. So we've just made our first enormous deposition into Chembl as well. Then you might also want all the structural data that we have. And just to give you an idea of how much structural data we are generating, our effort alone generated 6% of all new X-ray structures in the protein data bank in 2023. And just to give you an idea, that's we generated 600 structures and deposited 600 last year. All American synchrotrons only outdid us by a little bit, by about 50%. That's all other science in the United States. And we have a huge number of structures that are queued to deposit to the PDP this year, and really it's the PDB, the Fair Date Repository, that's the problem. So we have this rapid way of actually looking at these in this Fragalysis viewer as well. So it's an online viewer where you can download the structures, play with them interactively, superimpose them, etc. Of course, you might want to see what we've already published on these targets. Publications tend to happen for us a lot later than the data that's shared, but you can still see everything that's associated with each of the different programs. Really important in antiviral discovery is you want to make sure that you're making molecules that aren't going to easily be knocked out by a single nucleotide change, because as we're seeing with antibody therapeutics that target the outside of the virus, that's very easy to happen. We can target more conserved proteins, but we still need to worry about that kind of evolution and resistance. So we have various ways of doing this through deep mutational scanning and analysis of circulating variants. You can play with that data, and you can also visualize it interactively, looking at how, for example, these fragments engage either mutationally invariant or mutationally flexible protein residues. And there's an interactive link here where you can explore that. Here's just an example of one of our compounds engaging with that. And everything mostly is green, suggesting that we're not making any problematic contacts, but these ones are ones we might worry about. Then, of course, when you're starting any drug discovery program, you have to figure out what is the property of the pill that you're actually going to give to people. And this is informed by how you're going to deliver it, how you might do diagnostics to figure out whether you have this virus or not, and how you're going to use it in communities of interest or in a pandemic setting, or if you're using PrEP or PEP. So that this is done in consultation with other global bodies like the World Health Organization, the Drugs for Neglected Diseases Initiative, which is a part of our consortium. On the next stage, you then need, we build what's called a target enabling package, which is essentially a structure-based drug discovery kit that has everything you need to get started, including assays, plasmids to express the protein, protocols for purifying the protein, and X-ray structures to start with. And this saves possibly several years and shaving them off of drug discovery programs if you're getting started on that target that already has a target enabling package defined for this. This grew out of the Structural Genomics Consortium work, which has been very successful the last few years. And we're modifying this for antivirals, where we're thinking about how we can represent multiple members of the family as well. You can browse all this data interactively again before it hits the PDB. And then once you've got that package to start with, you turn the target product profile to what's called a target candidate profile, which is all of the objectives that the actual molecule needs to achieve in order to actually be persuade someone to spend millions of dollars to preclinically develop your compound to actually take it into humans for the first time. So there's things like it has to hit the targets in enzyme assays. It also has to actually get into cells and hit the targets there. It has to be soluble enough and have good PK. And then, of course, you have to worry about all of these safety issues as well. We're making these public because you don't normally get to see these. They're great educational tools as well, but they're really, here's the objectives we're working from for our discovery program. And other people might have different objectives, but it allows you to at least start with something you can modify. There's an assay cascade that's defined from that, which is just how you organize all the assays at your disposal to try to meet those objectives without going broke. It's often difficult to do that, but you try to put the expensive assays, especially the ones that involve animal pharmacokinetics at the end, and a lot of the earlier, cheaper ones where compounds can fail out much more easily at the beginning. Any assays that we developed ourselves go onto protocols.io, and we share those in a way as much as possible. The data for actual assays also is visualizable immediately, but ultimately ends up in Kemble. We have this for every stage from hit to lead optimization. And then finally, once we've completed a preclinical package, we're going to provide all of the data that's associated with the preclinical IND filing online as well. This is just an example of what that looks like. Sometimes a lot of data doesn't fit into this format, and we have nine total AViD centers that would like to share data and resources. So we also created this open forum where monthly we have 2 talks where people give 30-minute talks about some resource they generated, something they found that's relevant to antiviral discovery, and then can link the resources online. So we have this great library of talks. If you ever want to learn about any viruses of pandemic potential, I highly encourage you to go and take a look from it. Even industry has been participating in this, which has been phenomenal. And I'm at time, so I'm just going to thank the enormous number of people that have contributed to this from multiple different organizations and the NIH for funding the chemistry efforts. And thank you for your attention.
Thank you so much. That is just fascinating work, and that was quite the tour de force that you sped through. And I look forward to the discussion part because I know I have questions, and I'm sure our audience has questions. Please be sure to put those in the Q&A function because we'll have the Q&A after our next speaker, who is Dr. Daniel Harris, and we'll be talking about one of our Distinguished Achievement Award winners, GeoPIPE, reusing open data and letting data flow. But before I turn it over, let me tell you a little bit about Dr. Harris. He is an associate professor in the Department of Pharmacy, Practice and Science, and the Director of Clinical Research Analytics for the Institute of Pharmaceutical Outcomes and Policy at the University of Kentucky. So I will turn it over to Dr. Harris.
Thank you very much. It's great to be here. I will say this is very much a team science effort. Myself and Nick Anthony, our data scientist, and Chris Delcher, we have an interest in open data and how it applies to substance use disorders research. So we built a tool called GeoPIPE, and we've been promoting it through conferences. And we're grateful to be acknowledged as part of the DataWorks Prize. We are at the intersection of the geospatial domain and healthcare. So I tried to frame, what is essential between those 2 domains. So here's a large language model's opinion. I will show you some maps later on for sure, but also I'll discuss the importance of precision as well. That word is not a coincidence that ChatGPT picked up on that. That's an important word for this domain in particular. So let's talk about location data. The importance of social determinants of health is ever increasing as we realize that our location and where we are in the world plays a larger and larger role in our health and our health outcomes. There's evidence to suggest that zip code, for example, plays a larger role than our genetic code in terms of health outcomes. An important issue is privacy. Everyone has a right to medical privacy, obviously, and location is definitely protected by HIPAA. Often researchers don't really want an address for a patient, but really just what that address represents. They want to know facets of information about the location itself. For example, what is the poverty rate where this person is? Opposite to protected health information is open data. Open data is publicly available, typically with some type of license to use. Usually it's free to download, free to use for various non-commercial purposes usually. We have observed an increase in open data efforts, particularly around, again, I'm in the substance use disorders domain, particularly around medical examiners' offices, those people who are responsible for investigating and reporting deaths. There's some nuances from state to state, but death data is largely a matter of public record keeping. The ease of accessing those public records is what's going to vary state by state. And of course, some states, they are not public records. The open data examples I'm going to show in this presentation are not common. There's less than a dozen cities and counties across the US that make their data this openly available. And they are pioneers. They are not released typically for research purposes, although they know people like me will use them for what we want to do with them. But the primary goal is usually transparency to the public. So requests for information can be short-circuited to their websites and lessening the burden on their staff. The point of GeoPIPE is, again, that these data were not released for research. It's just to make using them and reusing them easier for researchers. So what is GeoPIPE? The goal is really to take raw open data that's publicly available online and to enhance it and to enable reuse as much as possible. We focus on substance use disorders and overdoses in particular. And our examples today are taken from Cook County, Illinois, who publishes a weekly, sometimes daily feed on their medical examiner's office websites. We do two important things. We add location-based data to enhance the utility of the geospatial aspects. For example, what census tract was this event occurring at? That's going to be our linkage to any socioeconomic data, such as poverty rates. We also create indicator flags for anything we might detect on the cause of death field. So drugs or substances, for example, if cocaine is mentioned in the cause of death field, we add a binary yes/no flag for cocaine. Opioid overdose in particular are chronically problematic across the United States. Last year, they contributed to about 75% of all overdoses. And for Cook County in particular, they are increasing in number over time. This is not adjusted for population, but, and also if you look at the 2020 data for Cook County, it's kind of outrageous due to COVID-19, which touches base back to the previous presentation. So how does GeoPIPE work? It's basically a custom pipeline that hooks up into APIs offered by the most common open data platforms. If you visit the Chicago data online, it looks like the bottom of the screen. This platform is Socrata. It's one of the open data platforms available. And there's APIs for pulling that data. We can programmatically pull it on demand, process it, and post the results. If you look at the primary cause of death field on the screen, you know, it's helpful that it's there, but many researchers just don't have the capacity to perform natural language processing. So part of our pipeline converts that text into indicator variables to tell you if a certain drug is present or not. So for example, if fentanyl is present, that fentanyl flag gets set to 1 as a true. So what's part of our pipeline? We geocode records. So we take the address data that's available there and we geocode to get coordinates. Those coordinates help us link spatially to Census Bureau geometries. We extract drugs from the cause of death field and do some spatial joins to create some additional fields too as well. So for example, did this event happen to occur in a park or not, or a public place? Those things are of high interest to researchers. So how does drug identification work? Currently, we use a string similarity technique, which captures typos, which do happen. Of course, you have to control the threshold for that string similarity to make sure it's not too loose and not bridging the gap. So for example, fentanyl is very similar to fetal, but those are much different words. So you have to control the precision there to make sure you're not matching inappropriately. But in our data set in particular, fentanyl is one of the most commonly occurring substances next to alcohol and heroin. In terms of pure number, again, these are not rates or adjusted for rates. We presented a paper on GeoPIPE at the American Informatics Association annual symposium, and we had some initial findings and using Chicago as an example, including talking about our geocoding process. Sometimes we were able to re-geocode records to get better results and kind of recover where those locations are. One thing to keep in mind with open data, it constantly updates itself. So sometimes you have to go back and backfill records to make sure you don't have any holes in your data. All right, so what's a use case? This is the slide that I really want to get to. This map shows Cook County. The white dots are opioid overdoses that occurred within 1/3 mile of a pharmacy. And the orange dots are those that occurred outside of that range, so greater than 1/3 of a mile. The blue areas are a 1/3 mile buffer zone for each pharmacy in Cook County. So why is this interesting? Well, 62% of overdose deaths in Cook County occurred within 1/3 of a mile of a pharmacy. I know not everyone on the call is familiar with substance use disorders research, but there's something called naloxone or Narcan. It's an opioid reversal drug, opioid overdose reversal drug, and it's highly effective if you have access to it. So the white dots are really missed opportunities for potentially life-saving intervention because they're so close to a pharmacy. The orange dots really indicate areas where resources maybe are not so easily accessible. So from a community intervention point of view, increasing naloxone where the orange dots are could yield positive change for that community. Another use case, this is a simple hotspot analysis, not adjusted by population again. So naturally, downtown Chicago is where a lot of the activity is going to be, and along the shorelines is what a lot of the activity is going to be as well. Basically, this shows you the biggest volume of activity that's happening. And again, recall that our open data source is constantly being updated, so we can constantly update this map too as well, and to see how things change over time. The whole process is automated using GitHub Actions. Our pipeline runs weekly. It's all hands off, it's all automated. So basically we tap into, we fetch data from these APIs that are hosting these open data sets. We do our geocoding and our spatial analyses, and we do our drug extraction. We join those to certain reference files to get additional columns, such as those census tracts that I mentioned, and those are going to be our gateways to things like poverty rates. And then we release that back on GitHub itself. So I showed you Chicago during this presentation, but we're actually doing this for about 6 or 7 different locations that have these APIs turned on for their open data sets. So for example, Milwaukee, a much different location than Chicago, but Milwaukee County has an open data portal too as well. What our GeoPIPE tool does, there's a configuration file where basically you can set your endpoints, what URL are you pulling data from, and what the primary data fields are that you need to do this type of analysis. So for example, what is the cause of death field for the medical examiner data? What is the location data? So by using this configuration file, we can kind of set parameters to basically pull data from any open data repository out there and run our pipeline on it. So we've kind of generalized the process. Chicago's the example here, but we could say the same story for each of these different jurisdictions that have their medical examiners data available as open data. I mentioned privacy is a big concern. Naturally, location data trips people out. And I mentioned there's kind of a big switch from protected health information from like the clinical domain, that the examples we talked about earlier, from the publicly available medical examiners data that is meant to be a public service. So those two are combating different ends. We have one very private, one very public. And I think in terms of what the science really needs is this validated methods. So one example is point obfuscation or point jittering. So if you have a point in space that you want to obscure where it actually belongs to, you add some noise and kind of shifts it around to a different location, so you have some imprecision in your data. But we have to be careful. By randomly moving that data point, you could be shifting important things that are important to study from a research point of view. For example, in this screenshot right here, this image, we're looking at poverty rates, and just by shifting your point by a few 100 feet, you can end up in a census tract where the poverty rates are wildly different. You can go from low poverty to extreme poverty, or areas of extreme poverty to low poverty. There's a QR code here for a paper that we published that attempts to fill in some of the methodological gaps for this type of thing, but I think we need to invest more time, more resources into developing out these methods as well. I know location data is typically hard to acquire. It just so happens because of the open data aspect of the medical examiner's offices that we've been working with. Those are actually open data sets because they're doing a public service. So I think we have a couple of bridges to gap before this location data is actually publicly accepted, but we believe this is one part of the roadmap to doing that. I do want to say I'm very grateful for being part of the DataWorks Prize, and it's great to see that open data projects are being acknowledged. When it comes to data reuse, data sharing, open data is, in my opinion, something that cannot be doubted with this respect to its utility for everyone. Thank you.
Great. Thank you so much to both of our presenters. And what an interesting, in the best way possible, contrast between your two projects, because we've got people-oriented projects and we have chemical-oriented projects. Please do have your questions in the Q&A, and there is already one in there, but I'm actually going to take a slight spin on it. John, if you would give me a little bit of flexibility, because the question acknowledges, you know, the benefits of having grant sponsorship of work, and I would be curious to hear from both of you about, and I know it came up in Daniel's talk as well, you know, let's talk a little bit about, everyone loves open science, but how do we make it sustainable? So I'll start with John, because I know he's been itching to answer this question, and then we'll go to Daniel.
Great. Yes. You know, the COVID Moonshot predecessor project actually had no funding at all. In fact, we ran a GoFundMe to try to raise funds for chemistry, because we just used whatever we had in the sofa cushions. For the successor project, you know, the US NIH decided to invest in $1.2 billion through HHS, the antiviral program for pandemics, in you know, getting ahead of the antiviral pandemic threats of viral families, but then decided to rescind that a couple of years later. So they've actually cut the program, and it will end 2 years early, meaning that most of the $600 billion public investment in this will just go in the trash, which is why doubly, you know, getting that data out there so that someone else can maybe pick it up and use it in the future is extremely important. So, you know, sustainability for the data sharing is also really challenging. The NIH has tried a few different efforts, especially with NIAID and the ODSET, to try to plug into the NIAID data ecosystem. There was a small supplement opportunity to get a little bit of funding for that, but that is, of course, very delayed. And it is unclear who would operate such a long-term infrastructure for doing this. So not only is, you know, the funding pipeline globally for therapeutic development for pandemics completely dry, and likely to be cut off at the knees, there is also nobody that's willing to step forward to you know, take ownership of sharing data that has been generated for these centers, and maintain some sort of ecosystem that would allow us to continue to, you know, connect up the data sources with fair repositories that will continue to be accessible in the future once these all wind down at the end of next year. So it's actually a real challenge. There's no real good solution at the moment. There are some funders, you know, small philanthropic funders that have tried to support the open source ecosystem, but I think this is a real critical need to make these things sustainable. I'm actually really in awe of what Daniel has done with automating things through GitHub Actions. Of course, these always are vulnerable to bit rot, but it's something that, you know, a single human in their spare time might be able to maintain, as opposed to a very complex infrastructure for doing this. So I'd love to hear from you, Daniel, too, about you know, how you envision this kind of structure for sustainability.
Thank you, John. And yes, I do think it's two things. One is just the engineering component, which is often understated how much effort that actually takes. I think using open source tools is great in terms of sustainability. But also, I think in terms of science, we need to start leaning towards automation and embedding that into the actual science itself. I think that part often gets overlooked as kind of in hindsight, wouldn't it be great if we had done blank? But actually, we should just put that up front and try to automate what the most we can. And I mentioned, our stuff on GitHub runs every week. There is some maintenance in that. That's not totally hands off. You do have to do due diligence to make sure you're not putting out trash data. You have to do some QA and some. So there's some overhead of ownership. And the second ingredient, I think, is really just, I think we need funding agencies to embrace infrastructure in terms of being able to build infrastructure and to build these types of tools too as well. I think there's hesitancy in what are we paying for in terms of resources? Is it a tangible resource, or is it an investment to something that's going to pay off in the future as well? So for me, it's two things. It's the engineering component, but also it's just the ability to fund that engineering component too as well. We did not have funding for this project, but we hope to one day.
Great. I appreciate both of your candor, because I think that is something we discussed with the DataWorks Advisory Committee quite a bit. It's these things are not free. We want to make them freely available. So I appreciate your candor. As I was listening to both of your presentations, one of the words that popped in, maybe I wasn't using ChatGPT, I was just using my own brain, but thinking about the trust that you each had to build with your communities, like in terms of antiviral development, and then also the trust with the public of how you are reusing the data. Could you talk a little bit about like how you build these agreements, as you're moving through these pretty awesome large scale projects?
Daniel, do you want to go first?
So I can make a quick comment about my project. So for our Cook County work, that was when some of the initial work we started doing open data. So we did partner with the Cook County Medical Examiner's Office and Mojay Mir, someone who we've been working with for a long time. They're an epidemiologist in the Medical Examiner's Office. And it was important to establish that relationship early. They actually became a co-author with us later on several papers, actually. But it was important to establish that trust of the medical examiner's office that we're not doing anything nefarious with their data too as well. We're trying to do good science. And I think when you establish those relationships, it becomes obvious, oh, actually, they're bringing a lot of attention to the good work that we're doing in terms of transparency and whatnot. And I think it's welcomed. So it's mutually beneficial. Yes, we do get to, in our case, use the data, enhance the data, and put it into multiple people's hands, but also it draws attention to them. They were truly a pioneer open data with respect to medical examiners data, which is typically from state to state can be hard data to acquire.
I'll just say that Daniel's point about the previous question is actually, fully applicable to how we approach this, where you have to think about the automation of data sharing from the beginning, and that's a big change in how people work. And so it did take a lot of work with the enormous number of folks that are producing, as you saw, a huge amount of data for our antiviral center, to work with them in a way that, they're just not saving everything for the end, right? Everything has to be visible not only across the entire organization, but externally as well. And that just breaks down a lot of barriers, both internally and externally. But it's a paradigm shift in how you do science.
Great, thank you. So, thinking about, again, trying to synthesize both of your presentations. Thank you for getting me that difficult task. What is the question that you wish people asked you about your work? Or what is the lesson learned that as we're closing out this symposium, what piece of advice that you have that no one asked you for? Of course, I don't know how to frame this question, but what would you like to leave out there? And I think you've already given such great nuggets about, planning ahead and thinking about what you can automate. What other things should, especially thinking about people who are just starting in this space and are realizing that these tools are available or there are these ways to streamline research, what advice would you give?
So the biggest question I wish people would ask is, why isn't there more of this in data sharing for drug discovery, right? Of course, industry has needs for keeping data private, but there's still a lot of pre-competitive data that could be easily shared. And in fact, both to industry and to a lot of nonprofits like Medicines for Malaria Ventures and Drugs for Neglected Disease Initiative are generating lots of open data anyway in pursuit of the discovery programs. Sometimes it just takes showing them a template for how this might work. I really like the checklist we saw earlier in terms of naming file names, for example, giving people a recipe that is a practical guide for them to use, and emphasizing that as a first-class product, and just showing people what a prototype looks like, I think can be a very powerful means for getting people to start thinking about how they might approach this.
So I teach the introductory data science class for our master's program in data science. And I often get asked, so here's a data set, like, how do I get started? And I think it comes down to getting familiar with it. And I think that's going to be driven by standards. So we have to have standards for metadata, we have to have standards for documenting data. All the data sets that I've shown today all have a really nice documentation component to them. And I think that gets undervalued as well. We have to have the ability to understand what we're actually looking at, or we're going to misinterpret something potentially. So my biggest take-home message is know your data when you're working with it. Otherwise, you might accidentally take some missteps and you won't even know it too as well. But also we should encourage people due diligence with documentation, metadata. Don't undervalue those things. Those things should come first, not as a post-mortem, an annotation of the data that you're actually releasing to the public.
I actually think that aligns really well with one of the messages we heard in the previous session about, you can think about the data sharing, but you are your number one user. So, know thy data is certainly terrific advice.